My name is Natalie, and I'm a postbac with the ADME Lab. There are approximately 7,000 rare diseases, but only about 5% have FDA-approved treatment. Fortunately, it's becoming increasingly possible to use gene therapy to treat rare diseases. One platform, called adeno-associated virus, or AAV, gene therapy, is particularly promising. AAV uses a modified virus to deliver a working copy of a gene to cells that have a defective one. We are focused on a disease called propionic acidemia, a genetic disorder in which the PCC enzyme is defective. This can be fatal. We wanted to see if we could use UPLC-MS methods to assess the function of PCC enzyme by quantifying the amount of product, Methylmalonyl-CoA. So we developed a UPLC method to measure the substrate, Propionyl-CoA, as well as the product, Methylmalonyl-CoA. Because Methylmalonyl-CoA can be converted to Succinyl-CoA, we wanted to measure that as well. This is hard because Methylmalonyl-CoA and Succinyl-CoA are structural isomers, and they're difficult to separate on the UPLC. However, if you look at these chromatograms, we were successful in resolving the two compounds. We have one peak for Methylmalonyl-CoA and a second peak for Succinyl-CoA. Now that we had a working method, we wanted to use it to assess PCC function in samples. Here we're looking at the formation of Methylmalonyl-CoA in mouse livers. At 0 minutes, none of the samples produced any Methylmalonyl-CoA. However, at both 30 and 15 minutes, the amount increased. You can see the wild type, which has 2 working copies of PCC, produced the most, whereas the mouse which had 1 working copy and 1 defective copy formed somewhat less Methylmalonyl-CoA. And the mouse which had 2 defective copies of the PCC gene did not form any Methylmalonyl-CoA at all. So we showed that our method was able to assess PCC function in mice with different mutations. Next, we wanted to see if we could use this method to assess the effects of AAV gene therapy. Similarly, we had a mouse with 1 working copy and 1 defective copy of the gene. And again, we see some formation of Methylmalonyl-CoA. One mouse which had 2 defective copies of the gene, again, no formation of Methylmalonyl-CoA. And then a mouse which had 2 defective copies of the gene, but was treated with the AAV gene therapy. Here you can see the formation of Methylmalonyl-CoA is substantial. So we were able to see the effects of AAV gene therapy and how it improved PCC function. This method was robust, free from interference, and useful for both in vitro and in vivo samples. Moving forward, this method will be used to assess toxicology as well as larger scale pharmacodynamic studies. Thank you.